Hello, my name is David Castillo, and I'm the CSO of BatchX. And in this video, I will show you how to run an RNA-seq differential expression pipeline in the platform, as well as take a look at the outputs that this pipeline creates. Right? I'm currently already logging into the platform, and we are in the tools and pipelines screen. Right? From here, you can type RNA-seq, and this will show you all the tools and pipelines under this category. For this case, we're interested in this pipeline, the differential expression pipeline. So I'm going to click on it, and this will take us to this other screen. Here, I can see all the documentation linked to this pipeline, which basically tells me what are the different tools that are being orchestrated by the pipeline, as well as the inputs that this pipeline requires, and some examples of how these inputs should look like, right? As happens with other tools and pipelines that we have in the platform, we already provide some sample data that users can test, right? Without having to upload any other data to the platform. And they will find this on usage. If we go into this screen, we'll see that there's this basic example that is the one that we are going to use. And we can run it just by click on this button. This takes me to this other screen where I can configure the execution. This has already been configured because I'm using an example. And from here, I can specify the name of the project or the execution that I want to give to this run, as well as the samples that I want to use and how I want to group the samples and do the comparisons. Right? So once all of this is being configured, it's just a matter of clicking on submit, and this will launch the pipeline in the platform. This execution has already been launched in the past, so BatchX will identify this and will not run again the pipeline. It will simply provide the same results that were obtained in the past. Okay, so if I click refresh, I would see that, okay, let me click again. Yeah, there you go. That now I have the details of the inputs that were provided, as well as all the outputs that the pipeline generates. Right? There are different outputs from the alignment files as well as other tables, but for this video, we're going to focus on the report that it generates. I've already downloaded this file and unzip it, so we can save some time and just take a look at how that report looks like. Right? So it's something like this. From here, we have different sections being included in the report. And the first one is quality controls. Right? This will give us an idea of how well the execution went, as well as if the quality of the samples that I used in the execution are okay to being used. Right? For example, in this case, what it's telling us is from the initial set of reads that I used or provided to the pipeline, how many of those were able to pass the different filters that were applied and were being used for the differential expression. We have, on average, 80 million reads, which is an okay number to perform an RNA-seq on human data, which is our case. We can also take a look at where these reads align. What you would like to see is that most of the reads have to align on exons, because that's what you're expecting when you're running an RNA-seq analysis. So in this case, again, this is a good distribution of reads, right? Most of them are being aligning in the exon, so we can consider that all the samples are okay. This other quality controls tells us how good are the genes being covered by the reads. We would like to see something like this, an uniform coverage along the whole extension of the genes, right? So again, these are optimal results and tells us that we can trust the analysis that we run. Everything that we are seeing here in these graphics is also displayed as a table, which the user can download either as comma-separated values tables or an Excel spreadsheet. Right? So once we have taken a look at the quality controls, we can move ahead and see the differential analysis and the results. In this section, we provide a series of graphics to also have a global view of how the differential expression analysis went, right? Again, this is other graphic that tells us if the samples are being clustered based on the group that we define, right? In this case, again, we're seeing that they actually behave as we will expect, right? And you can see that all the samples that were classified as untreated are shown together, and something similar happens with the treated, right? There's some level of variability within this group, but that's something that it's okay to expect, right? And this, by the way, is a PCA, which is a principal component analysis plot, and is something common that is being used in RNA-seq. Right? There are other graphics that we provide, which not only tells you how good samples cluster, but also how many genes were obtained or were identified as differential expression genes, right? We have MAA plots, box plots, and all of these are interactive plots. Once we have taken a look at all of this, you can also see a differential expression table that we generate using DSEC. And this basically will analyze each individual gene in the catalog and tell us if that gene was found to be differential expressed, right? You can sort, you can filter directly in this report, and you can also take a look at how these genes compare to each other between conditions, right? If we click here, this will take us to this plot, and we can see that all the samples classified as treated has a very low expression compared to the samples that are classified as untreated, right? So this is a gene that might be interesting for us because there's a very different expression between both conditions, right? All of this is an analysis that is being carried on per gene, right? But sometimes we are also interested in seeing if there are pathways or group of genes with the same or with similar function that are behaving in the same, having the same patterns, right? So that's what this other screen tells us. Here you can find pathways and gene ontologies on different classes, and it will tell you if any of these categories has differential expressed terms, right? For example, here we have pathways as defined as KEH that were found to be differential expressed because some of the genes included in these terms are also differential expressed, right? We not only provide the tables, but we can also inspect the plots, similar to the previous screen. And finally, we have a manual which explains all the columns, all the graphics, all the content that this report has, right? So this, in general, this is a general view of the RNA-seq differential expression. We have other types of pipelines for RNA-seq, but this is the one that we're analyzing here. And that's it. Thank you.